This topic is going to be on enzyme mechanisms. And so it's easy to get caught up with all the details of how an enzyme catalyzes a chemical reaction when looking at the mechanistic aspects. And there's experimental ways to help verify if a mechanism proposed is indeed at play. However, that's so beyond the scope of a general biochemistry course. However, I do want to emphasize some key principles involving enzyme mechanisms and looking at the serine protease family as an example of illustrating those particular facets of an enzyme catalyzed reaction. So the learning objectives that I'd like you to focus on are shown below. So let's start this topic of enzyme mechanisms by thinking again about the free energy of the enzyme catalyzed reaction and what the enzyme is actually doing to promote the reaction. So the best way to illustrate this is through the reaction coordinate diagrams. So let's look at the uncatalyzed reaction. In this case, we have two substrates, A and B, come together to form a complex, and then we have a chemical reaction occur to join these two molecules together through a chemical covalent bond. The energy of activation is the difference between the substrate energy level and the apex at the transition state. And this is your activation energy. Enzyme can catalyze a chemical reaction by lowering this activation energy in several ways. A, shown here, is an enzyme can lower the transition state energy by stabilizing the transition state, thus lowering your overall activation energy. The levels of the substrate and the products remain unchanged. Another way an enzyme can catalyze a reaction, so if we look back at the A situation here, is if we want to change the activation energy here, the enzyme could raise the levels of the substrates higher, but not affect the transition state so that the overall activation energy is lower, and that's shown in B here. Enzyme binding A and B, you can see it's higher energy than A plus B, but the transition state is the same level. So it kind of elevated A and B closer to the transition state and thus lowered the overall activation barrier of the reaction because of the higher energy of the substrates mediated by the enzyme. Part C does both. It will elevate the energy of A and B, but also lower the transition state. And this is a combination of the two. So usually enzymes are designed to lower transition state energies, and depending on its available binding energy, it can be utilized to bind substrates or the transition state, but it has to be able to recognize substrate, not just the transition state only. So it's usually a combination of the two in terms of the amount of available binding energy. You got to recognize the substrates through binding events, but you also have to have enough binding energy available to lower the transition state as the reaction proceeds. And so the reaction of an enzyme, or the mechanism of an enzyme, is to utilize binding energy to change the level of substrate and transition states in terms of their energy to lower the overall energy of activation for the enzymatic reaction. And enzymes are typically designed to bind the transition states the best, not their substrates. Okay, there's still some substrate binding, but they're designed basically to minimize that transition state energy. Notice in this reaction here, and on the left, if you're looking at delta G values here, if you wanted to calculate the delta G of the reaction, here it'd be a positive delta G because the products are greater than the substrates, and this would be an endergonic reaction. If it were exergonic, then A and B would be up here, and then A hooked to B would be lower. So you should be able to recognize these features of these reaction coordinate diagrams as being exergonic or endergonic as well. So I wanted to point that out if that wasn't obvious. So let's look at this binding of substrates. So we're going to look at a series of reactions here, and we're going to use organic chemistry to help us understand how an enzyme, just by binding two molecules and bringing them closer together, can result in rate enhancements. So here is a reaction with two organic organic molecules. This is an ester here, and this is your acetate ion, and we're going to form an anhydride in this reaction, and a leaving group is going to be this highly stabilized anion here that has a spectroscopic property, so we can measure the reaction of this anhydride formation through breaking of this carboxylic acid ester and forming the anhydride through the release of this anionic leaving group. And we can measure the rate of that reaction. This is uncatalyzed, and we can call that 1. In other words, the rate constant for this reaction with these two products, velocity equals K times the concentration of the acetate times the concentration of the ester, is going to be 1. All right. What if we hook these two molecules together through this this methylene bridge here? We have two of them. So there's freedom of rotations around these methylene groups for these molecules because they can swing around. But they have to be in proper orientation to facilitate this anhydride bond formation. If we have these hooked together and we form an anhydride, we're going to form a cyclic anhydride. That's a 6-membered ring, very stable thermodynamically. Still got our leaving group here, and we measure the rate of this. It went up by 1,000. So having two molecules try to come together in the transition state, they have to diffuse together to come together, okay, in the proper orientation. If we just hook them together, they don't have to diffuse together now. They just have to find the proper orientation. Having them hooked together and not diffusing together has increased the rate by 1,000. Okay? What if we limit the freedom of rotation by removing one of these methylene groups? And so now we only have one bond that will allow freedom of rotation. That's going to limit our conformational space and should enhance the reaction. And in this case, we form a 5-membered ring, and it does enhance the reaction by a factor of 200. So limiting degrees of freedom of rotation is another way to enhance a chemical reaction. And if we limit that orientation even further through a bicyclic compound here, and we have these two substituents at the equatorial positions on adjacent carbons of this ring system, that's going to limit freedom of rotation altogether. And so you're just basically bringing these things together in proper orientation, and that gives you a rate enhancement again of another 200 or so. So now look where we have come from two molecules that have to randomly collide to being in close proximity, exactly positioned properly to promote the reaction. It's now 5 times 10 to the 7. So that's a huge enhancement, and that's just by bringing two things together. That's what enzymes get some of their rate enhancement from, is this ability to bring two things together and orient them properly in the enzyme active site. That is the proximity effect of enhancing the chemical reaction. So recall that we were using the Km is the concentration of substrate in order to get 1/2 maximal velocity for an enzyme catalyzed reaction, and that good enzymes, efficient enzymes, operate at low substrate concentrations where the Km values are low. Poor enzymes would have to have higher concentrations of the substrate. And so this has to relate to how the substrate is bound and recognized by the enzyme. And so the best way to think about that is to look at the reaction cordon diagrams once again. So if we look at an uncatalyzed reaction where we have the substrate, again, we're looking at the single substrate situation, but you can imagine the same thing for two substrates as I shown earlier in the first slide example of the reaction cordon diagrams. But let's just focus here on this enzyme binding to the substrate, okay, just the substrate binding component. Here's the uncatalyzed reaction, and on to the right is the enzyme catalyzed reaction. Now notice the E plus S has to combine, so there's a little energy barrier initially to allow two things to come together, because again you're freezing out some rotational motions and disorder in the system because you're bringing two things together. So there is a little energy barrier there, and you're forming an ES complex, and then you're going through the transition state of S, and then you form your products. And so for the enzymatic reaction, we're lowering the transition state by the enzyme binding to the active site in the active site with the transition state. So the ES double dagger is the transition state in the enzyme active site that's going to be of lower energy than the transition state of the substrate without the enzyme being present. So there's a lowering of the transition state energy. Now, in terms of binding, available binding energy, how is it utilized to bind substrate? Well, it could be one of two ways. You could just bind it enough so that you get it out of solution into the enzyme active site, and then use the remaining binding energy for conformational changes in the enzyme as S approaches the transition state to promote the chemical reaction and lowering of the transition state energy. Or alternatively, you can use more binding energy to bind the substrate more specifically and tighter, and that's going to lower the energy of the ES complex. But if you lower it too much, most of your binding energy is going to be utilized in the binding step, and there won't be much available conformational change in binding energy available to bind a transition state. So you want the enzyme to bind the substrates, but not too well. If it's too well, then your activation energy will be much larger, and you're not going to help catalyze the reaction. So you want to bind it enough, but not too much. And so that's why this Km value helps you think about the ability of the enzyme to bind the substrate in terms of its efficiency. It's kind of related to this energy level here in the reaction cordon diagram. It's not a KD because, again, it's binding, but it's utilizing some of that energy as it approaches the transition state. So you have to think about it in terms of being able to recognize the substrate but not bind it too well. Okay? So that's what we want to think about there in terms of the enzyme. We need to be able to bind substrates but not too tightly. It's not like an antibody binding an antigen. It's not catalyzing reaction, it's just recognizing something. So we want high binding specificity. But in terms of an enzyme, we want it to recognize substrates but not too well, because otherwise it just forms a potential well here, and it won't be able to help stabilize the transition state enough to allow the activation energy barrier to be small enough to catalyze the reaction. So this is play between binding substrates and the transition states. So we don't want it to go 100% one way or the other, because if it just bound the transition state, it wouldn't bind the substrate very well. If it just bound the substrate, it wouldn't bind the transition state very well. So there's got to be a balance, and how that balance is achieved dictates the Km and Kcat values. In terms of the mechanism of binding, there are two types of binding models. One is the lock and key model of substrate binding, and the other one's the induced fit. The lock and key model is such that the enzyme has an active site such that, or binding site, such that the substrate fits in it perfectly. And the substrate just comes in and it binds, and then, or two substrates come in and bind, and it promotes the reaction to occur to form product. Whereas the induced fit has the enzyme in a certain conformation, and when the substrate binds, there's a conformational change in the enzyme as the transition state is approached, and then the product forms. So there's some movement, some dynamic movement that's occurring during the from the binding event to the transition state. And so the induced fit model is pretty much how enzymes catalyze reactions. There's a conformational change approaching the transition state to help stabilize it. And then once the transition state has been achieved and the chemical reaction occurs, then product released then is promoted through another conformational change. And so the induced fit is the one that best explains enzymatic activity. Lock and key would be more of just rigid binding without any conformational change. But proteins are very dynamic, and they're not rocks. And so usually they're not going to just be super rigid. And you can probably get some induced fit even just upon binding as well for some enzymes. So these were the two pervasive models. And the lock and key, just binding something, again, without any movement, is pretty much how you define what no movement is. But there's usually going to be some movement involved. So the induced fit is helps explain this approach to the transition state and stabilization. And evidence for that is shown here in yeast hexokinase. So part A here is just the unbound form of the yeast hexokinase, and the active site is kind of shown here. Okay, it's fairly open, and you need the active site to be available to the solvent because substrate's got to go in there and bind. So it can't be closed off. It has to be open. Some enzymes exist in a closed state, and they have to be activated to open up. And so sometimes enzymes are inactive because their conformation closes the active site. You may need an event like phosphorylation of a serine residue, for example, to cause a conformational change in the protein to open up the active site, and now it's ready to catalyze the reaction until that phosphate has been removed by a phosphatase. So there's some control of this active site through conformational, or I should say covalent modifications, or interacting with another molecule or protein can also induce a conformational change. And so you have to have it open, okay, to allow substrates to come in and bind. Water is going to be lining this as well, so you got to wait for some, displace some waters so you can get some hydrogen bonding and some recognition occurring as well, okay, to get some other molecules out of there. So there is some dynamics going on, but once the substrate comes in there and starts binding and water starts to leave, some of these features, as shown in the induced fit model, will start hydrogen bonding and binding to the substrate, and you'll have a conformational change in the enzyme itself. And this kind of moves down this direction, and you can see where the substrate is bound. And so that's best illustrated if you were to take your slides like a flip book, and you can just toggle back and forth, back and forth, back and forth. I'm not going to do that here in this presentation, but I'm going to illustrate that just by going from the next slide, which will show the picture of the unbound E form, and then it'll show a picture of the ES form. Again, these are experimentally determined structures that have been done. Often it won't be necessarily the substrate, or if it's a two substrate reaction, it may just have one substrate just bound. So if A has to bind before B, and this is a two substrate reaction, yeast hexokinase, this would be your A binding first. So there's a conformational change. Then B would have to bind, and then you can do the chemical reaction. So I'm going to show the unbound form, and then I'm going to flip quickly to the bound form, and you can see the protein will make its change. And you can do this on your own slides, back and forth, back and forth. So let's take a look at it here. Unbound, bound. You see the change? That's evidence of an induced fit conformation. So let's look at these active sites in a little more detail. So these are going to be interiors that are going to be allowing substrates to bind. And there's groups in there that promote binding and catalysis. These active sites usually don't have water in them, but sometimes they need a key water molecule or two because they're important for the chemical reaction or substrate recognition. In other words, water is not necessarily 100% not involved, but it could be involved. But it just depends on the reaction being catalyzed. Of course, hydrolysis reactions, you need water. So they would have to be involved there for the hydrolases enzymes. There's also, in terms of substrate recognition, you want there to be good contact between the enzyme and the substrate to promote binding and to freeze out rotational, vibrational motions to limit the degrees of freedom like we see in the proximity effect. Okay? If we look at amino acids lining active sites, a lot of them are nonpolar. Those nonpolar residues of the side chains for the amino acids that are nonpolar, like leucine, isoleucine, phenylalanine, those types of amino acids will be providing a hydrophobic environment. And with water being excluded, this allows for a different dielectric constant in the active site that can promote electrostatic effects that are much more prevalent and stronger in the active sites of enzymes than in aqueous solution, where you have water and their interactions with these charged molecules and dipole alignments. Okay? So the key thing is that there are key polar and charged amino acids in the active site that will be involved in binding of the substrates and the reaction mechanism, in addition to the hydrophobic environment that provides a more electrostatically favorable active site for interactions. So the dielectric constant there being altered without the water being present. And so hydrophobic, but still polar, key polar groups for binding, specificity, and catalysis. So that's the structure, organizational structure of enzyme active sites in general. If we look at specific amino acids that are involved in a lot of enzymatic reactions or enzymes, you can see that the pKa values can alter drastically based on their free amino acid values. So we spent a lot of time in other topics talking about the pKa values of the free amino acids, and I did mention at that topic that these pKa values can change based on the environment in which the amino acid is in, the solvent, or in this case the enzyme active site. So in an enzyme active site, we just have to consider those side chains that have an ionizable group. And in free amino acids, those groups have a amino group that's protonated and a carboxylic acid group that's deprotonated at pH 7. But in an active site, these alpha amino groups and alpha carboxylic acid groups are involved in peptide bonds. So there is just the side chain itself. And that side chain here can have a different environment based on what it's in. Now the terminal end of the protein will be a little different, as we talked about too, in terms of the N terminus and C terminus. But I want to focus on just the side chains in terms of the values. They can range quite a bit in their value because of that dielectric constant that's present in an active site that has most of the water excluded, if not all of it, and the ability of the hydrophobic environment promoting electrostatic interactions and binding events to allow these pKa values to be different because you're in a different chemical environment, okay? And one of the, again, don't worry about memorizing these numbers, but just understanding the active site of the enzyme can promote pK lowering effects by interactions with active site groups that aren't possible outside of the active site. And an extreme case, and we'll come back to this with papain, is a protease, and it's a cysteine protease, and it has a pKa value of 4.2, whereas cysteine side chains have pKa's around 8. It's 4 orders of magnitude lower. That's because of the environment in which that sulfhydryl is in. It can easily lose a proton, almost as a carboxylic acid group would, because its pK is around 4. So that has to do with the active site. So the geometry, the chemical groups that line the active site causes chemical properties to change a little bit in these side chains because of that environment. Where in aqueous solution, those values are different, okay? It's all through binding events. So let's look at some of the generalized mechanisms. We don't have time to go through this in detail. I don't expect you to memorize any of this, any of these examples. I will show a couple examples to emphasize key features as a way to see that, yes, these mechanisms do prevail. You will also see some of these mechanisms throughout the course when we want to emphasize a particular reaction mechanism for a particular enzyme that's very important in a particular pathway. But you will not be required to memorize details of these mechanisms, but you should understand the chemical principles involved, okay? The one mechanism that we'll cover here that is required for any general biochemistry course is called the serine protease mechanism. And that one's pretty straightforward, so it's not too bad. It involves a lot of acid-base chemistry, which you should have a pretty good understanding of. But right now I just want to focus on these chemical aspects here of these enzyme catalyzed reaction mechanisms. They usually involve nucleophiles and electrophiles. So remember, nucleophiles are usually negatively charged, and they like to attack electrophiles that are usually positively charged. So it could have partial negative or partial positive charges, and they work together, these nucleophiles attacking electrophiles. And so different examples of nucleophiles can be shown here. Here's a hydroxyl group that has been deprotonated to form a negatively charged oxygen. That's a wonderful nucleophile. Remember, the stronger the base, the greater the nucleophilicity. So a hydroxyl group as a base has a pK of like 18. So you pulled the proton off, it's a super strong base. It wants to pick up a proton. It's a great nucleophile. So there's that relationship with nucleophilicity and basicity. The sulfhydryl group of cysteine can also lose a proton and become negatively charged. It's a good nucleophile too, but not as good as the hydroxyl because it's not as strong as a base. A carbon can lose a hydrogen and become a carbanion. That's a good nucleophile. The lone pair of electrons on an amine is a good nucleophile. The lone pair of electrons on the side chain of histidine, that's in an sp2 orbital, not in resonance with the ring, is a good nucleophile. And then if water loses a proton and you generate hydroxide ion is a great nucleophile. It's like this one up here. All right. Those are examples of nucleophiles, and those can be promoted in enzyme catalyzed reactions in active sites. Electrophiles are usually a series of reactions in which you have a double bond usually present at some point. A carbonyl group, because you have a polarization of the carbon-oxygen bond, delta plus, delta minus, a nucleophile like R with a lone pair of electrons, like an amine, for example, would like to attack that carbonyl group. Instead of a double bonded oxygen, you can have a double bonded nitrogen and form these amines or shift bases, and those are also prone to nucleophilic attack at the carbonyl carbon. A phosphorus of a phosphate group is also a good electrophile. You can see that instead of a carbon-oxygen bond, it's a phosphorus-oxygen bond, but the same things apply in terms of the polarization of that bond and its delta plus value there. It's prone to nucleophilic attack. This one's shown here in terms of a proton being reacting with a lone pair of electrons on a nucleophile to form a hydrogen connection, covalent bond to the molecule. But in this case, I think of this more of a protonation step, not a nucleophile electrophile reaction. It starts to get a little confusing there. It's like, what's the nucleophile? This lone pair. What's the electrophile? The proton. Yeah. But we usually reserve that as a protonation event, acid-base chemistry, as opposed to nucleophile electrophile chemistry. So the ones we're going to be focused on are going to be the ones with these double bonds, okay? In addition to nucleophiles and electrophiles, there's also the ability to form carbon-carbon bonds as well. And so you can take something that's a poor nucleophile, but it can become activated and become a better nucleophile. So you can deprotonate it usually. That's going to be involved in there. And carbonyl groups provide the polarization needed to change neighboring attached carbons to become more nucleophilic. So for example, you have a carbonyl group here, and here's your delta plus delta minus, a good nucleophile on its own, but we can change in terms of B here, we can cause it to become deprotonated, and now you have a lone pair of electrons here that can add a component that can be used as a nucleophile. So this carbonyl group can help make something more acidic and make it more nucleophilic. With an amine, the same thing can happen. And also another way to make this more electrophilic is to polarize this carbonyl oxygen bond even more so. A metal ion can bind to the lone pairs of electrons on the oxygen to promote this bond to be more polarized, making your carbonyl carbon more delta plus. Or you can protonate it. In this case, it shows an acid here, but it really didn't protonate, or through hydrogen bonding interactions with a proton with an enzyme active site amino acid residue can help polarize this bond even further to make it more prone to electrophilic attack. And then once you have some of these compounds that are carb anionic but resonance stabilized, you can form carbon-carbon bonds to like aldol condensations or Claisian condensations to form carbon-carbon bonds. And indeed, we do see these mechanisms in enzyme catalyzed reactions and metabolism. Last one here, you can also look at decarboxylation reactions of beta keto acids like amino acids. For example, we looked at the decarboxylation of the arginine being the one that has huge rate enhancements, and we talked about how it takes like maybe 4 billion years for the half-life of that to be decarboxylated, but with the enzyme you can have 1,000 per second. So basically you're losing carbon dioxide in the process. And with the beta keto acids, this would be your alpha carbon here, like with your amino acids, but it has a beta keto group. And so we'll see this a lot in metabolic enzymes where you have decarboxylation reactions, and you'll form these beta keto acids. So these are just some examples of the diversity of the chemistry that can be provided by enzymes. Again, I don't want you to memorize all these details. Just be aware that when you're looking at these pathways, and we're talking about their enzyme catalyzed reactions, these are the mechanisms that will be at play depending on the substrates being utilized by the enzyme. So the next category are acid-base reactions. And so we spent a lot of time talking about acid-base chemistry. And so let's consider these reactions in regarding enzyme catalysis mechanisms. So these reactions depend on the amino acid side chain groups that are in the active site. And they either can act as bases or they can act as acids. Remember, bases are proton acceptors and acids are proton donors. So if we think about pH 7 and what the state of that amino acid would be, typically the groups that could pick up a proton would be your histidines, aspartic acid, and glutamic acids, because they've already been deprotonated at pH 7, and so they will have a tendency to pick up a proton. But that's not always necessarily the case in an active site. It could be that they could be protonated. It's possible, okay? But on average. The acidic groups would be the ones that would be protonated and could give up a proton. And histidine, again, is one of those that's the pKa is such that it kind of hovers around that physiological pH. So you always have some that's protonated, some is deprotonated, and may be favored one way or the other in an enzyme active site. Lysine is usually going to be protonated and can donate a proton. Arginine, not so much, because its pKa is very high, and it's going to just basically think of it, and plus its charge is delocalized too. It's going to be tough to, that's why the pKa is so high. It's tough to deprotonate that one. So we'll just think of histidine and lysine as these groups that could provide a proton if needed. But if glutamic acid is protonated, it can give up a proton too and be an acid as well. So it just depends on the mechanism, right? So if we have base catalysis, what's happening there? There's an extraction of a proton from the substrate to activate the reaction. In other words, a base in the enzyme active site is pulling a proton off from the substrate, and now it's creating a conjugate base form of that substrate that's important for the enzymatic reaction. Okay. Water can also be mediated in this process if it's involved, and it would generate a hydroxide ion, which could be in the mechanism as well. So again, water could play a role. Acid catalysis happens through direct protonation. In other words, you add a proton to the substrate, and that's needed to activate the reaction. So this interplay of proton donation and proton removal is key in enzyme reaction mechanisms. Not all of them, but many of them. And so the one I'm going to really focus on, the serine protease, is a series of these acid-base reactions. Covalent catalysis is another one in which the enzyme itself is covalently linked to the substrate or some part of the substrate in a manner that the enzyme now has a new form. It's like an acylated form. It's a modified form. And it has to be removed, and it's usually through hydrolysis by water coming in to knock off that group to recreate the free form of the enzyme that can go through another round of catalysis. That's just one example, but there are others where the enzyme itself is covalently modified in the process of the chemical reaction that involves this strategy that's called covalent catalysis. So that's covalent modification of the enzyme through part of the substrate, not the whole substrate, but part of it. So if it's transferring something to another group, it may take the group like a transferase reaction. Part of A is removed, stuck on the enzyme covalently, and then it adds it to B, for example. The chymotrypsin mechanism that we'll talk about goes through covalent catalysis but it's also mediated by acid-base reactions as well. So some of these mechanisms don't necessarily have to be in one category, but they can be in multiple categories depending on what the mechanism is. The last part is the effect of pH, and this should be no surprise because we went through detail study and doing problems in which the pH of a solution can affect the charge or the protonation state and the population of those states based on pKa values of that molecule. And so the same thing occurs here in enzymes, specifically if there is important residues in the active site that need to be protonated or deprotonated, the pH can have an effect on that as well. And so the effect of pH on reaction rates are one of those conditions you test to see what your mechanism of your enzyme is. So typically things are going to be around pH 7, but what happens if you run it at pH 6, a buffered solution at 6 or 5 or 4? So you do a variety of different pHs and you measure your enzymatic Michaelis-Menten type measurements to see how your enzyme is behaving, and then you plot enzyme activity here as a function of pH. So you do it at different pHs and you get the enzyme activity. This could be your Kcat over Km, for example, at a given pH, and you can look at your enzymatic rate constant as a function of pH, and if you see something that looks like this, that's telling us that at low pH, whatever this pH value is, it's working pretty well, and then as the pH goes too high, it starts to drop off, and then all of a sudden the enzyme's no longer active. There's no enzymatic activity. And if you see a transition like this and go to the inflection point here, you'll often see that the pH is going to be hovering around a possible pKa value of amino acid that may be important in the active site. In this case, this would indicate that since the pH is around 4, we could postulate that perhaps an aspartic acid or glutamic acid were important in this enzymatic reaction, because we get this nice inflection point. We're like titrating that particular residue and it's affecting the enzymatic catalytic efficiency of that enzyme. And we don't know for sure, but that's what we would postulate based on this type of data. Okay, and we would need other experimental evidence like a structure that we could look at the active site and see if there is an aspartic acid in there. And so there's a lot more to prove that, but this does give us some insight if acid-base chemistry is important. Okay, and so at low pH is protonated, that's what's needed, and once you deprotonate that thing, it's got a negative charge, it's not working anymore. So maybe it's participating in an important hydrogen bonding event, or maybe it's donating the proton to the substrate that's needed in the active site for the reaction to occur. So you get some insight, but it doesn't answer everything. So you have to do a lot more experiments to really pinpoint a detailed mechanism. But don't forget, pH is important. So let's look at pH dependence for papain. We mentioned that papain has a cysteine that has a pK of 4.2. So let's look at its pH profile. So this looks a lot different than the example in the previous slide, where we have no activity at low pH, and then activity starts to increase as the pH goes up. We go through some kind of transition here, and it starts to level off, but then the activity goes down as we keep going to higher pH, and then eventually drops off. So we have what we call a bell-shaped curve here. And the inflection points on the rise of this curve indicates that if this group is protonated, nothing's happening until you start to deprotonate it. And then when it's completely deprotonated, the activity is great. And it goes for a while, but then as you keep raising the pH, you're starting to deprotonate something else, and now its activity is going down. So it indicates here that this residue must be protonated and has a pK of around 4.2, and then the activity will go up as it's more deprotonated. So we want it completely deprotonated in the apex here, and then there's another group up here that's protonated, but if you start deprotonating that, it's going to cause the activity to go down. And so we have a transition, or the transition here at the apex of activity has this group deprotonated and this group protonated. And that's indeed what we can tell, that there's a group here that has to be deprotonated and a group here that has to be protonated. So this may be acting as some kind of nucleophile or required for removing a proton, and this one is involved in some kind of proton donation or hydrogen bond or something that's required. And we don't know what those residues are. You'd have to look at a crystal structure and make mutations to see if your bell-shaped curve would change to try to find these residues. But other evidence is required to identify them. But you know there's at least two of them with these two pKa values. And when it was researched and looked at, it was cysteine that was responsible for this amino acid residue, and histidine for this. These are way outside their pKa ranges, and that's because the environment of the enzyme promotes the deprotonation of the cysteine sulfhydryl of cysteine and keeping the histidine protonated for this reaction mechanism to be promoted for that given substrate. So the unique environment of the enzyme active site can change these pKa's quite a bit. So the pH here would have to be between 4.2 and 8.2 if you want some kind of enzyme activity. If it drops too low or too high, you're protonating something that shouldn't be protonated if you're down here, or you're deprotonating something you shouldn't be deprotonating here. So pH, again, very important for whether the residue is protonated or not. The final aspect I wanted to cover regarding enzyme catalyzed reactions is in terms of transition state stabilization. So if it hasn't been clear up to this point, enzymes stabilize transition states to promote chemical reactions. Okay, so there's very tight binding between the reactants and the enzyme in the transition state. Okay, so the enzyme lowers the activation energy barrier by lowering transition state energy. And Linus Pauling recognized this and said that enzymes are molecules that are complementary in the structure of the activated complexes of the reactions that they catalyze. And so that is to say that if you were to look at an enzyme active site, it is designed to bind this high energy transition state. So if you could make a molecule that looked like the transition state, you would have a tight binding molecule that that enzyme would recognize and would form an ES complex, that S being your transition state molecule, so tightly that that would be the best binding molecule that you can make for that enzyme, because enzymes are designed to bind transition states. So if we look at an example here of dihydroxyacetone phosphate and the chemical features of that, we have a carbonyl group, we have a -2 charge phosphate group, we have a hydroxyl group here, we have a couple of protons off of that alpha carbon. And if we look at the transition state that's involved in the reaction of this particular enzyme, in this case is triose phosphate isomerase, is that there is a proton being pulled off of this alpha carbon. There's going to be electrons going to be dumped in here to basically form a double bond and an OH group that's shown in this enol-diol intermediate here. So you can imagine pulling off a proton here, the electrons go here to form a double bond, this pi bond breaks, oxygen has the electrons and it picks up a proton, and you got an OH group. So you have this ene, because you got a double bond, diol, because you have two OH groups on there. It's like a dialcohol, two H groups. So it's called an enediol intermediate. And so that's what's happening here. You have a proton abstraction, and then you have a proton donation. It's a rearrangement of the electrons. So there are no electrons being lost. It's just a rearrangement. And so if we look at the transition state of what that would look like, you're basically looking at a deprotonated alpha carbon with those lone pairs of electrons distributed across the alpha carbon, the carbonyl carbon, and the carbonyl oxygen as a negatively charged species. So you have to have groups that align the active site to help stabilize this charge that's being induced on the molecule from a neutral charge to a negative charge. And so if you could design a molecule that had a negative charge over here, you probably have a good binding molecule that would work. So if you look at the same reaction here and look at a molecule that may mimic this, you can imagine if we have a phosphate here, keep that the same, keep the methylene group the same, but change this into a carboxylate, acidic group that would be deprotonated. It looks like that a little bit, right? It doesn't have the OH and the H on it, but you can see that charge delocalization is in exactly the same spot. And so this potentially could be a good binder to the enzyme. And we'll talk about inhibitors in another topic, but this molecule cannot be catalyzed in a sense where it's going to form this enediol intermediate, because it can't. It's chemically not able to form the enediol intermediate, so it cannot be reacted upon by the enzyme. But because of the chemical features of this molecule, it can bind to the enzyme active site very well and inhibit the enzyme from being able to bind the dihydroxyacetone phosphate and catalyze the reaction. So it would mimic the transition state, and since it has structural features by nature similar to the substrate, it would bind to the enzyme active site and bind so strongly, much more so than the substrate, because it has this extra negative charge on this end of the molecule that's lacking here, that it would cause the enzyme to be inactive because it would be occupying the space the dihydroxyacetone phosphate would normally bind. So it would inhibit the enzyme. And we'll talk about how we measure enzyme inhibition in terms of molecules that bind non-covalently and cause the enzyme activity to go down. This is the whole concept of a drug, where we make drug molecules that mimic what the enzyme normally does to fool it into binding it. So if you were going to design a drug, you don't want the drug to look like the substrate. You want the drug to look like the transition state. In order to understand the transition state, you must know the enzyme mechanism in order to figure out what the transition state is, and you'll know what groups are involved, and you can design a drug that's going to mimic the transition state and bind with high specificity, because many enzymes are family members and they bind similar substrates. And so if you just design the drug based on substrate recognition, it can bind a lot of different enzymes and really screw things up. The specificity comes in stabilization of the transition state, and that's where your best drugs are designed based on transition state analogs, as they call them. It's all related to this concept of enzymes bind transition states. In fact, Linus Pauling speculated that antibodies, since they bind antigens so well with such high affinity, he said if you could make a transition state and raise an antibody to that transition state, it would catalyze that chemical reaction. And indeed, the case that was shown, that like a transition state analog like this, you could have an antibody recognize this 2-phosphoglycolate, and it would catalyze this dihydroxyacetone phosphate conversion, just like triose phosphate isomerase would. It wouldn't be as catalytically efficient, okay, but it still could catalyze the reaction relative to no catalyst being present, And some of these antibodies could be very effective in chemical synthesis in providing the stereochemistry and regiospecificity needed to make enantiomeric pure compounds. And so these antibodies were called catalytic antibodies, and it was an experimental realization of what Linus Pauling postulated in terms of the enzyme active sites being complementary to the transition state, thus promoting this concept of transition state stabilization. In our example of triose phosphate isomerase, I wanted to show you the actual binding of this 2-phosphoglycolate to the active site, and so you can see it's forming these hydrogen bonds with key residues like histidine 95 and glutamate 165. Again, you don't need to memorize the details, but this is showing you that indeed it is the case where you know something about the mechanism and something about the transition state of the chemical reaction. You can design very potent inhibitors of the enzyme, and this is the showing you experimental evidence of that. To finish out this topic, we're going to look at one family of enzymes called the proteases, and we're going to look at the mechanism of proteases, specifically serine proteases. So here is a diagram showing you the relationship between these different proteases and their activation. Proteases are enzymes that are secreted in the small intestine to help us digest our food. And they're made in the pancreas, but if they were active in the pancreas, they would start digesting the pancreas up, because they're basically, proteases are used to break down peptide bonds. In other words, they hydrolyze peptide bonds. So they take proteins and they help break them down into peptides, and there's other proteases that take the peptides and break them down to the amino acids, and then we can utilize the amino acids in synthesizing new protein, or we can use that as sources of energy as well to make 3-carbon precursors to be involved in carbohydrate metabolism, so, and use it for energy. So we have these a number of proteases that assist in digestion, and so in this case, making them in the pancreas, we don't want them active, so they exist as a zymogen, which is an inactive form, and they have to be modified in such a way to become active. So again, it's a way to regulate the enzymatic activity, turning it on when you want to, but not when you don't. In this case, it actually has some extra amino acid residues that have to be cleaved and removed in order for it to become active. Okay, so we're not going to go into the details of a lot of this, but I just wanted to show you it's the way of having it being inactive and becoming active. Okay, and so these little green areas are showing the activation aspects, and here's trypsin, one of the serine proteases that's involved in a lot of activation of the different enzymes. Okay, and so green is active, and then you can look at the diagram. Again, it's not an emphasis here, this cascade, to memorize it, but just know it's a process that once it gets started, it starts activating these other proteases. Okay, and the zymogens have different names. So if it's trypsin is the active form, trypsinogen is the zymogen form because it has the ogen name on it. So if you study nutrition and things like that, you may be talking more about these zymogens in terms of digestion procedures. But just wanted you to, I'll just use an example here of how the activation occurs. We just don't have time to go through all the details of this process, but just focus on the biochemical principle. So we're going to focus just on chymotrypsin, and chymotrypsin is shown here in the figure. And we have different forms of the chymotrypsin. In the process of this molecule here on part A, this is the zymogen form, so that's really chymotrypsinogen, all right? And you see certain residues are highlighted. There's these yellow residues, green residues, and red residues, okay? And you can see between A and B, the orientation, the red ones seem to be in the same position. The yellow ones have moved, and the green ones are gone. So what happened here? There's some different structures. So some of the structure looks the same, like right here looks the same, around here looks the same. But over here, much different. Something happened. Something removed these green ones, and this yellow one was way over here, it's moved over, now it's over here. So this is the active form of your chymotrypsin. This is the zymogen form that's inactive. So if we look at the sequence and see what happened, that's shown here. You have peptide bond cleavage events. So the amino acid residue 13 and 14, the peptide bond there was cleaved. The peptide bond between 15 and 16 was cleaved. The peptide bond between 148 and 149 was cleaved. And between residue 146 and 147 were cleaved. So these intervening sequences, 14, 15, and 147, 148, these dipeptides, there's two of them, those are in green. So those have been cleaved out. So what cut those? Trypsin and chymotrypsin itself. It's called autolysis, autohydrolysis of these peptide bonds. So trypsin and chymotrypsin itself causes these two groups to be removed. And so when you lost these two dipeptides, there was a huge conformational change in this part of the molecule, swinging this side chain here into the active site to help promote the binding of the substrate and catalysis. So in the zymogen form, the binding site isn't realized because the two key residues are far apart. But getting these dipeptides out by activation by trypsin, then chymotrypsin itself, causes this to rotate into position to form the binding site for the substrate and allowing these catalytic residues to do their job, which are in red, to promote the reaction. So this is an example of how chymotrypsin goes from the zymogen form to the active chymotrypsin form. Now we're going to look at how, when substrates bind chymotrypsin, how do they cleave proteins into peptides. So those three red residues are these three residues here. Now the numbers will vary based on the organism and the protease, but all serine proteases have this catalytic triad. They have an aspartic acid, a histidine, and a serine. Again, I don't care about the number, that's just the residue number for that particular enzyme, but the residues themselves, you should know what the catalytic triad is. Aspartic acid, histidine, and serine. And knowing the role of each residue in the catalytic mechanism, we're going to go into that. So here's a diagram it was taken from the structure of the enzyme, showing you the proximity there and their location along the backbone. We can strip that away and look at the interactions in the hydrogen bonding that's occurring between these residues. And so you're looking at an aspartic acid here that's deprotonated, acting to interact with a hydrogen on histidine here to promote a hydrogen bonding interaction. And then you have a lone pair of electrons on the other nitrogen of histidine, so this histidine has to be deprotonated in order for this to occur. So you're looking at aspartic acid interacting with this hydrogen through a hydrogen bonding interaction of histidine. And histidine, in this case, is a lone pair of electrons on this nitrogen sp2 orbital, is hydrogen bonding to this hydroxyl group. In fact, it's strong enough to pull it off, causing this carb-oxygen-hydrogen bond to break and to form an oxyanion here on this serine hydroxyl group. So the pKa lowering effect is at play here, because the serine now can lose a proton because of this hydrogen bonded network with aspartic acid in the proper position of histidine to abstract that proton. Now you have a good nucleophile here the serine that's negatively charged, as I talked about in the previous slide, it can now as a good nucleophile to attack a carbonyl carbon of a peptide bond. So this is the catalytic triad to activate this serine to nucleophilically attack the carbonyl carbon of peptide bonds. That's the basic thing I want you to keep in mind. Okay? pH is going to be important in this reaction if you did a pH profile, kind of like we did with papain, is that if you start to protonate the aspartic acid, it can't hydrogen bond because it has to be negatively charged. And you start protonating the histidine, it's not going to work either because you have to have it deprotonated. So if you can do pH curves, you would see the effect of the protonation of these particular residues at low pH. So low pH, everything would be protonated, and then the carboxylic acid would be deprotonated as you increase pH up. Then eventually the histidine would, and then you'd see an effect in the increased rate at higher pH. So if you want to lower the activation of chymotrypsin, lower the pH to around 4, and it won't work because you'll be protonating these key groups. Want to get it active, get it up there at 7, and you'll be fine. So when we actually make these enzymes for some of the work we do in biochemistry, we'll make a stock solution, but that's an acetic acid solution. So the pH is 4, so everything's protonated. The trypsin is fine. It doesn't. It's not active. It's not going to cut itself up. So the next series of slides will be looking at the reaction mechanism of chymotrypsin. So I've tried to explain that in terms of details. Your textbook goes through it as well. There's many videos online if you want to watch those too. I'm going to focus on just a figure and try to focus on the key components. And I don't want you to memorize this, but just think about what's happening. Looking at the figure, do I know what the electrophile is? Do I know what the nucleophile is? What is the role of the catalytic triad in this? Do I understand it in this particular part of the mechanism? What is happening? What is the substrate binding? When's the product being released? When's water coming into this mechanism? So that's what I want you to think about, the chemical principles that are happening, not histidine, whatever number it is, or anything like that, okay? So this information is here for you to help you read it, look at the figure, go back and forth. I'm not going to read that here in the voiceover, but I'm going to focus on the actual reaction itself. This is for you to read later. All right? More information on the different intermediates, and I'll talk about those too. Intermediates are not transition states, okay? They exist momentarily in the catalytic process to form products from the substrates. They can be detected by, say, for example, NMR. So there's something that exists at a time we can measure it. A transition state is fleeting. We can't measure transition states. Too fast, too energetically unfavorable. It's like, boom, it's there, it's gone. We can't measure that. But intermediates we can, and I'll try to emphasize that as well. Okay? So here's the mechanism of chymotrypsin. So I like this diagram because it takes each step and gives you detailed information. I'm not going to read it. I'm just going to focus on how I look at it and how I'd like you to think about it as well. So let's look at the free enzyme form, the E form, before anything is bound. Where's your catalytic triad? You should be able to recognize it. Here's aspartic acid. I don't care about the residue number. It's deprotonated. It's hydrogen bonding to the histidine. Nitrogen that has a hydrogen atom attached. Okay, there's your hydrogen bond there. Here's your histidine side chain. Here's a lone pair of electrons. That's hydrogen bonding to the hydrogen on the hydroxyl side chain of cysteine. Okay, so there's my catalytic triad. What's happening down here? Well, there's a hydrophobic pocket that's important for chymotrypsin residue specificity of substrates. Okay, here are two glycine residues. It's got NHs, so those must be amide hydrogens that are helping to provide hydrogen bond donors that may be needed down here in what's called this oxyanion hole. Oxyanion is negative, so maybe that's going to be hydrogen bonding to these amide hydrogens to help stabilize whatever that oxyanion hole is. And then here's the active site. Okay, makes sense. Here comes our substrate coming in. This binding pocket likes this phenylalanine, perfect size, hydrophobic. Let's see how that fits in there. This color-coded here, so my guess is the peptide bond is going to be shown here. The C double bond O NH, this peptide bond is going to be cleaved. So you're going to have your two products color-coded there. Okay, let's see how it happens when it binds. Boom, it's bound. Okay, so there's that binding pocket. So you got phenylalanine in this hydrophobic binding pocket. Okay, the rest of the molecules just different amino acids. So part of the protein will be bind here, and the rest of it will be sticking out. It's just these residues will be locked in this active site. So there's your binding hydrophobic binding pocket for your hydrophobic side chain. I wonder if other hydrophobic side chains will bind there. Phenylalanine fits pretty well. Okay, and over here on this other amino acid R, I guess it doesn't care what R is, so the specificity is here. That carbonyl is kind of close to these hydrogens, so it must be doing some hydrogen bonding here. It looks like there's a hydrogen bond that's positioned right there on that carbonyl group before anything happens. So that adds to specificity of binding. And then we have our catalytic triad shown here. And so what's happening next? Well, what's going to happen next is that upon substrate binding, things rearranged a little bit, because you can see that these groups moved a little bit, induced fit to form a hydrogen bond here. And this is going to promote the deprotonation of the serine hydroxyl group by this lone pair of electrons on this nitrogen here to make this more nucleophilic and attack this carbonyl carbon, because we know that carbonyl carbon is delta positive on the carbon, delta negative on the oxygen, a good electrophile. So now we have a good nucleophile in close proximity with a good electrophile. So we're promoting that reaction through deprotonation. And then we have a covalent bond formed between the oxygen and carbon, and breaking of the pi bond to form the oxyanion, the negative charge on the oxygen. That's going to go through a transition state, which is not shown. But what is shown is the tetrahedral intermediate that is short-lived that we can measure. And that exists. So you have this tetrahedral intermediate. In other words, this carbon has 4 bonds attached to it. One of them is a covalent bond to the oxygen of the serine. So now we got a molecule attached to the enzyme. So the enzyme has been covalently modified. The histidine has the proton on it, so now it's positively charged. Okay, this intermediate is stabilized through these hydrogen bonding interactions. Notice now there's 2 of them. Initially we only had 1. Now this other one can participate because of the conformational change in the enzyme and the carbon going from sp2 hybridized to sp3, so it changed the geometry in the interaction. So now you have it stabilized through this extra hydrogen bond, but it can do one of 2 things. It can be here, it either breaks down and goes back this way, or we can move forward. So let's move forward. So what happens next? Well, these lone pairs of electrons go back down to reform the pi bond, but instead of breaking the carbon-oxygen bond going back this way, we're going to break the carbon-nitrogen bond of that peptide bond. And as that bond breaks, the electrons from that bond are going to be obtaining this proton that was originally on the serine hydroxyl from the histidine and become protonated, so you have an NH2 group here. See? Notice the H is from that. And now you have NH2 of that cleavage product, and that dissociates away. As soon as that forms, it falls off. It's gone. And what's left now is this covalently linked remainder part of your protein through this serine hydroxyl or oxygen, I should say, through this ester linkage. So we have this acyl enzyme intermediate, as they call it here. This first product has left. We have an acyl enzyme intermediate. So how do we get back to a free serine hydroxyl like over here at the beginning? Well, this is an ester, and so let's cut it with water. So water comes into the active site. We never consider water being a substrate, but it is. And since the water concentration is so high, we don't really think of it as varying in concentration, and so we don't consider it as substrate B. So that's why we don't have it as substrate B. And the mechanism here is called a ping pong mechanism, and so it's because of this acyl intermediate is so reactive with water, and it happens very quickly. So it's more of a ping pong mechanism that we're looking at here, as opposed to a 2-substrate. Even though it is 2-substrate, we don't really consider water as substrate. So suffice it to say, there's room now for water to come in and to attack this acyl intermediate, and the same mechanism happens. As water comes into this active site, just kind of diffuses in there. The nitrogen on histidine has been deprotonated because it gave up its proton up here to the leaving group of the broken peptide bond, and it will pull off the hydrogen off of water to create hydroxide ion, which is a good nucleophile that will attack the carbonyl to form another tetrahedral intermediate that's stabilized by the amide hydrogens through hydrogen bonding in this oxyanion hole. Then this intermediate either can go back or it can move forward. If it moves forward, then it reforms the C double bond O, and it's going to break the carbon-oxygen bond that's attached to the serine side chain, and the electrons that are formed through that bond cleavage is going to pick up the proton from the histidine side chain that was protonated to regenerate our free serine hydroxyl group that's now hydrogen bonded to the histidine group, and then you still got your hydrogen bond with aspartic acid that kind of holds that in place. And now we have our carboxylic acid side chain now of our other part of our protein. And again, in the active site, there's a proton there, but that will lose a proton as once it diffuses out and gets into solution. And that's our second product release. And then we're back to the beginning where everything's ready to go to bind another substrate. So what I want you to know about the mechanism and the role of these residues. What is the catalytic triad? What's the role of each one? What's it doing? What's it participating in? What is the binding pocket, the hydrophobic pocket? What's the oxyanion hole? What stabilizes the oxyanion hole? That's what I want you to think about. And so it's pretty cool, right? All these different things we talked about: hydrogen bonding, hydrogen donation, picking up a proton, losing up a proton, all very important in terms of how enzymes recognize substrates and catalyze reactions. In terms of the different families, they all have this catalytic triad, but each family member has a different specificity: chymotrypsin, trypsin, and elastase. And that's based on this substrate binding pocket. So the substrate binding pocket for these enzymes in this family are designed differently. So chymotrypsin, for example, cleaves peptide bonds on the C-terminal side of amino acids that have aromatic and large hydrophobic side chains. So it can account for large aromatic and hydrophobic side chains. So not only does it cut phenylalanine, but it can cut tyrosine, for example. All right. Some of the leucines and isoleucines can probably bind in there and fit and have some affinity as well. Trypsin, notice its binding pocket is a little bigger, longer, and it has a negative charge at the bottom. There's an aspartic acid down there that's deprotonated. So it allows for charge-charge recognition. So trypsin cuts at lysine and arginine residues. So you can see an arginine here. It's going to have a nice positively charged side chain, and that's going to electrostatically interact with that negative aspartate there, and some hydrogen bonding as well. And you have a nice binding pocket to accommodate such a large residue. Lysine would fit in there very well. And trypsin has a specificity to cut at lysines and arginines. Elastase, another family member, very small little binding pocket, and it's lined with valine and threonine. And so it can't accommodate big side chains like tyrosine or arginine, but it could account for smaller molecules like alanine or glycine. And so it'll cut at the very small side chains. Okay? The C-terminal side relates to the peptide bond that's being cleaved relative to the C-terminal end of the protein or the N-terminal end of the protein. So it just depends how it binds. Some proteases do N-terminal side, some do C-terminal side. All of these do C-terminal side because the C-terminal group is the one that's going to be in terms of the product that is released last. It has a carboxylate on it, so it's the C-terminal end. So it just depends on the way that the molecule reacts. But the specificity is based on recognition of the side chains of the substrates. The mechanism is exactly the same as in chymotrypsin, except the substrate specificity is different based on the binding pocket. So modulate the binding pocket, keep everything else the same, you got a different enzyme with different specificity. So if you were to design an inhibitor of proteases, you'd want something to bind like the transition state, or like a tetrahedral intermediate would be a great one because that's very close to the transition state, right? It's not the transition state, but pretty close. So this molecule here, this boronic acid compound, it binds with a different specificity here. We can have it such that when it reacts with the serine hydroxyl and it changes from sp2 to sp3 to bind to the oxyanion hole, this bond here, this borate ester, cannot be attacked by water. So it becomes a covalently linked enzyme that no longer can lose this molecule that was attached to it. So in other words, the enzyme reacted with this molecule as though it was the substrate, formed this ester, borate ester, but the borate ester cannot be attacked by water, and so it becomes an enzyme that has basically killed itself, a mechanism-based enzyme inhibitor. So these mechanism-based enzyme inhibitor have to be very specific for the enzyme because it has to bind with high specificity, in addition to the fact that the enzyme actually reacts with it as though it were the original substrate, but in doing so, it does it in such a way that it gets covalently modified that it cannot break down and go back to the free enzyme form, and so the enzyme kills itself. And so they used to call these suicide inhibitors, but they're now called mechanism-based inhibitors because it's based on the enzyme mechanism. So if you were to make a drug, yes, target the transition state, but make one that's a mechanism-based inhibitor that the enzyme has to act upon the inhibitor, and in doing so modifies itself and it no longer can catalyze any reactions anymore, that's going to be your best drug. So I just wanted to emphasize this concept of mechanism-based inhibitors, that you can design an inhibitor that the enzyme thinks is a substrate and does a chemical reaction with it, but modifies the enzyme that it can no longer be a free enzyme to catalyze any more reactions, and it's done. So this outlines some important aspects of the mechanism of chymotrypsin. I don't want you to memorize this, but I want you just to think about it in terms of your knowledge of the mechanism, and this stuff should make sense in terms of what's happening. Can you read this and see that in the diagram that I showed you from Leninger? Can you look at another diagram of a protease mechanism and understand what's going on? What is the nucleophile? What's the electrophile in that particular step? What's the role of the histidine in that particular step? Is it acting like a base and pulling off a proton, or is it acting like an acid and donating a proton? That's the kind of thing I want you to think about in terms of the mechanism here. So the problem sets on this section, the problems, talk about mechanism a little bit and have you think about it in terms of some other proteins like other proteases, but the concepts are the same. They may have a little different residues, but they're acting in a similar fashion based on this catalytic triad mechanism: proton donation, proton abstraction, creating a nucleophile in the active site. May not be serine, it could be cysteine in the case of cysteine proteases, for example. But understand the role of what's going on in terms of what's the nucleophile, what's the electrophile, what's acting as a base, what's acting as an acid. That's what I want you to take from that. Can you see a diagram and understand it? Or understand what the catalytic triad does? Can you fill in parts of it based on knowing the mechanism of serine proteases? So work on the problem sets, work on the mastering questions, and read the textbooks and watch some other videos to have a better understanding of mechanism. This is the only mechanism you'll be responsible for having an understanding of. And throughout the course, we'll talk about mechanism of some enzymes to help us understand their regulation and how they catalyze reactions. Because again, that gives us some insight as to how they're regulated, but you're not going to be responsible for knowing any of these mechanisms in detail.